We are going to speak about the ELISA, that is enzyme-linked immunosorbent assay. So this is a very basic diagnostic technique used to find the viral diseases, that is HIV, the herpes, and the many more. So what exactly the ELISA does is, here we have antigen and antibody interaction. And this antigen antibody interaction, we are conjugating it with an secondary antibody again, which is the secondary antibody is having a enzyme along with it. And this enzyme of the secondary antibody reacting with the substrate to give the colored product. So in the short, it is nothing but we have an enzyme linked antibody which is going to indirectly give the proportionality of the antigen present in the given blood sample. So how exactly do we do it? So first we'll learn about the principle of the ELISA, that we have antigen antibody interaction. So here what is happening? We know that the antigen has the affinity, the antibodies have an affinity for the antigen. So this affinity binding will happen. So let's say we have extracted a serum sample from the patient's blood, that is HIV positive person. So his serum sample will have a certain antigens, which is giving positive results for the HIV. Let's say he has certain viral particles of the HIV, glycoproteins or the proteins, all of it. So we have an antibody which is able to bind to this antigen. So we are making this antibody to bind to this antigen. Now that interaction between the antigen and antibody, we call it as a primary interaction. And this is a very specific in nature, the interaction. So the specificity, the antibody has a specificity to bind to the antigen. Now, once already bound, you can see in this picture, we call the second one as an enzyme labeling. So what is exactly enzyme labeling is, we have the primary antigen bound to the primary antibody, our first antibody. Now this primary antibody is made to react with the secondary antibody. Now our secondary antibody is something special. So why is special? Because it has an enzyme bound to it. Now as we know, enzyme has its own work as it does. It has a active site which is able to bind to substrate, which gives the product in the later stages. So now this enzyme is conjugated to your antibody. That antibody itself, we call it a secondary antibody. And this secondary antibody is bound to the primary antibody. Now, during this reaction, the end of the reaction, we call it the signal amplification, where exactly we are going to add the substrate. Let's say our secondary antibody is a galactosidase enzyme. We will have a substrate which will have a galactose, which will break down the galactose. So now, since my enzyme is a galactosidase, now I will add the substrate which will give the galactose, and then I will give the chromogenic effect which will give the color. Now if you see, the secondary antibody is not directly but indirectly it is connected to the antigen. So whatever antigen is present in the sample, it is bound to the primary antibody, and that antigen bound to the secondary antibody. The secondary antibody has an enzyme. Upon giving the substrate for that enzyme, it gives the product, and that product gives the chromogenic effect. Now think this in terms of the multiple antigens present in your sample, and the multiple primary antibodies binding to those antigen, and then the secondary antibody binding and giving the product. This is how exactly ELISA works. The end product is a colored component. The color is coming from nothing but the enzyme and substrate reaction. So here, antigen antibody interaction is not giving color. The antibody and secondary antibody interaction is not giving color, but the enzyme linked to the secondary antibody is giving the chromogenic effect upon the reaction, enzyme substrate reaction. So that is why entire concept we call it as an enzyme linked immunosorbent assay. That means the antibody is linked with an enzyme to give this kind of a result. So we have one of an assay picture over here. We can look at it. So antigen followed by the primary antibody, the secondary antibody, and the secondary antibody is conjugated with the enzymes. And then we have a substrate for appropriate enzymes. So we have a multiple enzymes. We look at those enzymes. See here, the peroxidase enzyme is the source is given. It is from the horseradish. Now, for the peroxidase enzyme is it is a redox reaction. So we will give the peroxide. So in the redox reaction, at the end of the day, you get the product, and that product is now again made to react with the chromogenic effect. The chromogen is added to it, and that gives the color. So that color is directly proportional to the antigen present. So let's say a person has a very dilute amount of antigen, the color will be dilute. So if the antigen is higher, so the color will be a bit of a intense. The dilutions are done in the microtiter plates as well. So we'll come to it later. So we have another enzyme called alkaline phosphatase. Uh, this also used in the enzyme substrate reaction. So we have a beta galactosidase, which is extracted from the E. coli. So again, it breaks down the galactose. So here, then we have a urease enzyme. The breaking down of the urea happens here. So these are the enzymes which is used for the reactions of the enzyme-linked immunosorbent assay. Now, what are the types of ELISA do we have? We have a direct ELISA, we have indirect ELISA, and we have sandwich ELISA. So what is direct ELISA is, now you take the sample of a patient uh, antigen, they say. Now, this antigen is directly made to react with the secondary antibody. There is no involvement of the primary antibody. Now, this secondary antibody is conjugated with an enzyme, and once the reaction happens, the interaction between the antigen antibody happens, then we will make the enzyme to react with the substrate. We will add the substrate later, and then the substrate will give the color. So here, since the primary antibody is not present and it is directly involvement of the secondary antibody and antigen, we call it as the direct ELISA. Second technique we have is an indirect ELISA. So indirect ELISA also we can have two types again. Where exactly first is where exactly antibody is already coated to the plate. So let's say you want to find out the uh, you want to have a detection for a disease, let's say. So you know which disease you want to detect. Let's say you want to detect some tuberculosis. So you have antibodies which is presenting for the tuberculosis, which is able to bind to the tuberculosis, which is already plated in your coat. So that is coated in your plate. So that microtiter plate is coated. So later what you do, you micropipette your antigens, which is extracted from the serum sample. Now, this antigen will bind to your already coated antibody in your plate, so which is already fixed. Okay, so that interaction happens. After this, we are going to add the primary antibody. Now, again, the primary antibody is again able to bind to the antigen. There is a specific interactions here. That interaction occurs after the primary antibody interactions. We have the binding of the secondary antibody that is happens to the your primary antibody. The secondary antibody binds to the primary antibody. So we know the the initial antibody is already fixed to the plate, so coated to the plate, and then we have an antigen bound to the the antibody, coated antibody. Then we add the primary antibody and then the secondary antibody. At the end of the reaction, we will add the substrate which will react with the enzyme and give the product, and the color will be formed. And this color is read through the flow cytometer. So this is how exactly indirect ELISA works. So we are using here multiple antibodies to get the results. So this gives a quite a precision than the uh, direct ELISA method. So now finally we are going for the sandwich ELISA. What is sandwich? So this name itself says what exactly is a sandwich. We have two sides antibody between antigen, like your two sides bread, and in between you have your salads. So here you have two sides antibody in between antigen. So that is what exactly sandwich ELISA. So here what happens? the plates are coated with the antibody, and then we are adding the antigen. Now we are going to add the secondary antibody followed by that. So first we have antibody coated on the plate. That is our primary antibody. And then we are going to add the antigen that is from the patient serum sample. And then we are going to add the antibody, secondary antibody. So this is how exactly goes. So the primary antibody binds to the antigen and then the secondary antibody binds to the antigen. So this will make the sandwich of the two antibodies. So this is how exactly at the end of the reaction, again, the enzyme binds to the substrate. We are going to add the substrate and the enzyme reacts to the substrate. So indirectly, the color will be formed. So in all the reaction, you understand it's a secondary antibody conjugated with the enzymes which gives the chromogenic effect. So, but you know, all the secondary antibodies bound to the primary antibody or it is bound to the antigen. So, indirectly, it is giving you the results how much of the antigen is present. So, this is how exactly the types of ELISA works. So, we will see the procedure of the direct ELISA. The procedure antigen is already plated to the sample is already plated, and for this sample plated, first very important is washing the microtiter plates or the plates every time, because freely not bound antigen will be there in the place and that will create issues, that will bring the non-specific interactions. So reduce that, we need to have antigen bound first and then we need to wash. Why washing is that? The antigen which is not uh, tightly bound to the plate will be washed. Otherwise, that will give the non-specific interaction. After that, we are again adding the secondary antibody. We are allowing to have the interaction between the antigen and antibody. After this antigen antibody, we are having a buffer wash again, so that the whichever antibody is not bound to the antigen should be washed out. Otherwise, random antibodies will give the results which is not appropriate. So only the interacted antibody should give the result. So once we are incubating for the secondary antibody and antigen, we will keep it for the incubation and wash it after certain time. So finally, after washing, we will add the substrate. Now this substrate will react with the secondary antibody which is bound to the antigen. Right? This secondary antibody has the enzyme. So it gives the signal at the end, so which could be read by the flow cytometer. So this is how exactly direct ELISA works. It's a coating, locking, and detection. Now we will go for the indirect ELISA. So indirect ELISA, so we have a microtiter plate over here. So here we already have an antigen coated well, where exactly antigens are fixed to the well, and then again, once we have added the antigens or fixed the well, we have to wash the well with the buffer. Why do we wash it? Again, the random antigens which is not bound to the titer plate or microtiter plate will be giving the results. So we do not want that. So we will wash the microtiter plate with a buffer. Second, we will add the antibody, the primary antibody we are going to add. Now, this primary antibody will interact with the antigen and that antigen antibody interaction will happen. Again, not all the antibodies binds to the antigen. Certain antibody which is not bound to the antigens will be washed after the incubation. So all of the free floating antibodies which is not bound to the antigens should be washed out at the end of the incubation. So wash with a buffer. After that, we are going to add the secondary antibody. Now this secondary antibody will be interacting with the primary antibody. Again, at the end of the incubation, we will be having the incubation for the interaction between the two antibodies. So they bind to their specific sites. Once their specific binding occurs, we will wash out the secondary antibody which is not bound to the buffer using the buffer. So finally, at the end of the reaction, we are going to add the substrate. So now this substrate will react with the enzyme which is linked to the secondary antibody and gives the results. The very important part is here. Just imagine having the secondary antibody not bound to the primary antibody and freely floating in the buffer in the microtiter plate at the end of the time. they are able to give the results. So very important to wash your secondary antibodies. Okay. So now finally we have sandwich ELISA. Again, sandwich ELISA, we have antibody coated in the microtiter plate. So the coated antibody is there. Now, let's say you have taken the patient sample, very highly used technique sandwich ELISA comparatively to indirect ELISA or the direct ELISA because very precision results it gives. So once you add the antigen, it will bind to the antibody. Now this antibody is directly bound to the antibody. So now here your sample might contain multiple number of the antigens. Like you might have some viral fever symptoms. You might have a different microbes or the different viral, but the antibody specificity is very specific in nature. So this antibody will bind to the specific antigen. Now, this antigen is made to react with another antibody, which is a secondary antibody. There are two types of sandwich again. There's a capture method sandwich ELISA. Now, I will directly skip the second antibody itself. I'll consider as a secondary antibody. Now, this secondary antibody has the enzyme linked to it, and now that second antibody binding to the antigen here, sandwiching the antigen between the two antibodies. Now, after that, we are going to wash it with a buffer. Once we wash it with a buffer, again, we are adding the substrate and the substrate gives the results. Now, sometimes it is very important. What happens is the substrate is directly does not able to give the results in the colored form. So whatever substrate is converted to product probably reacts with some other product and give the color. So we use TMB also in this reaction, which gives the blue color effect. So the substrate directly will not forming product will not be able to give the color. So color chromogenic effect will be given once the product is formed. Okay. Now, what are the reagents materials used in ELISA? So microplate, we see here microplate. These are 96 well microplate, that is 12 into 12 in size. Then we have antibodies. We require two types of antibodies. One is primary antibody and the secondary antibody. So that is the antibody we use. And then we have substrates, substrate specific for the enzyme. So if you're using the peroxidase, you need to have a peroxide as in substrate and further on. And buffers. There are two types of buffers we use. One is a washing buffer and then one more is a blocking buffer. So we'll come to it, what is blocking buffer in the later slide. So washing buffer is used to remove the randomly or the free antigens or antibodies which is not bound to the or does not have the interaction. But exactly blocking buffer is used where the primary antigen and antibody is having interaction and to make this interaction stronger. So that is what exactly the blocking buffer is used. So washing buffer is randomly uh, removes the freely floating antibodies and antigens, where exactly the blocking buffer will makes the interaction strong. So this is a typical. This is from the Fisher, Thermo Fisher. We have taken the how exactly ELISA is performed in the different scales. So you can see here we have 96 wells, which is 12 into 12. Now this wells you can plate with the antigens, plated with the antibody already, fixed antibodies plated to it. Now, once this is fixed, you are going to add the sample, that is your dilution sample, which is with the buffer. That sample is nothing but the serum which you extract from your patient's blood or any of the fluids. So once you do, here you can use the multiple people sample at the same time. Like you do not need one titer plate is not for one person sample. You can use multiple people, like for the detection purpose. So once you add your sample, you will be keeping it for the interaction, that is antigen antibody interaction with the sealing the. So incubation happens for 90 minutes, followed by at the 37 degrees Celsius. So once this incubation happens, after that we will wash with the buffer, washing buffer for preferably. and it is washing happens not once, it happens twice or thrice. The better the washing, so that we will not have freely floating antibodies and antigens in the microtiter plate. So then we add the secondary antibody. Here we are using biotin labeled antibody. So we are going to add that. Once we add the biotin labeled antibody, so we are having now again 60 minutes incubation, which is at 37 degrees Celsius. We are covering the microtiter plate. Once is that done, we are using the buffer for washing, and then we are adding the uh, substrate, that is TMB substrate here we are using here, which gives the blue color product. So once the substrate is added to all the wells, we are it is going to give the blue color within 10 to 15 minutes, nearly 10 to 20 minutes approximately. Once that reading comes, each titer plate is made to read under the flow cytometer. So it gives the differentiation. Let's say one person might be having a more number of antigens, or the other person will be having the less number of antigens. So that dilution between the people could be read in the flow cytometry. So I have spoken about what is specific binding and non-specific binding. So if you are having a blocking a buffer we use for the interaction between the antigen and antibody, the affinity of the binding to the antigen is stronger. Once it is bound, once you add the antibodies, the binding affinity is stronger. So it will bring the specific binding. Where exactly without the blocking buffer, uh, we have the antigen antibody interaction weaks, and we do not have the all the antibodies washing away. So that's why we get the non-specific interaction, which gives the poor results. So what is the advantages of ELISA? A very high sensitivity test, and we could diagnose majority of the diseases under the ELISA kit, and multiple samples could be analyzed in the ELISA, and very simple, straightforward. But the limitations of ELISA is it's very tedious process. It's a very manpower consuming, it's a labor consuming process. Think about pipetting 96 wells and washing every time, again adding your antibody, secondary, primary antibody, again you're washing, again you're adding secondary antibody, again wash. It is not like each well has to be added with the microtiter the pipetting. So all this takes a lot of labor, and it takes almost half a day. So that is why it takes a lot of manpower. This is how exactly it works. And if there is a non-specific binding or any of the interference between the molecules, that will give the poor results, and that is one of the limitation in ELISA. How exactly ELISA is used? Used for the quantitative and qualitative process. Quantitative is how much of the sample is present. But the qualitative is whether it is present or absent. The positive and the negative results exist. Yes, thank you.